Merci beaucoup, Anne. I will though switch to English. I'm confessing here, I've spent quite some time in France when I was about 13 years old as a high school student in Boston, in Bordeaux. So this is how good my French can be. So listening to the talks, I appreciate that I can understand about 30% of what's said. So this is why I'm switching to English now. And I'll speak very slowly in my English. So my interests are to understand genomic variation and by understanding and studying genomic variation, to learn about mechanisms in biology and more particularly disease mechanisms. And my laboratory to achieve this is using both computational biology as well as experiments. So I'm a laboratory combining these two different approaches. Our main interest is, and the mission of our group, as I say, is to understand a very particular form of genetic variation in our genomes, so-called structural variations. These are large differences between our genomes, large chunks of DNA that differ between people, and compared to single nucleotide polymorphisms, the other form of genetic variation we learned about much in the prior talk, these forms of variation occupy a bigger part of our genome and hence are assumed to be important and responsible for quite a number of diseases. Autism being one, and autism, though not being mentioned here, being one. Down syndrome, of course, being another syndrome that is broadly linked to a duplication of a chromosome or to partial duplications of that chromosome. And several other diseases that are heritable being linked to this form of variation, as well as cancers that arise, as we all know, due to variations that form during our lifetime somatically. And most of my talk actually will be on structural variations that we acquire during our lifetime that will then lead to cancer. To understand these variations in the genome, a big part of my research has been the development of methods, of approaches that allow to detect deletions, insertions, and inversions in the genome, which is not as easy as one may believe, because these events can be very complex. They often are in regions of the genome that are showing repeats and are hence hard to sequence. Nonetheless, we've embarked on this. Much of the methods development we've been performing was done in the course of a project that is known as the 1,000 Genomes Project, in which we were involved in an international collaborative effort to map genetic variants, including structure variations, in several populations across the globe. And I won't go much into detail with regard to this. I'm showing you, I'm relating here to the work of the 1,000 Genomes Project, as it had a very principal effect and influence on how we're performing our research. We've learned through this project how to deal with large sets of DNA sequencing data and how to interpret them. We've also made a number of interesting discoveries with the data from 2,500 human genomes recently. For instance, identifying 200 different human genes that appear to be entirely missing in some individuals, not all at once, but some of these are missing in some people, indicating some sort of redundancy in our genome that's probably a bit higher than previously assumed. And we've also learned about where structure variations are located in our genome and got some insights, or at least could develop hypotheses in how they form. They often are clustered together, so we see them near each other in so-called hotspots of the genome, where they're also enriched for disease regions. I now would like to focus on, as I mentioned earlier, somatic variation in our genome, specifically cancer. And I should acknowledge a very valued colleague, Stefan Pfister, a young medical doctor from the German Cancer Research Center, who I was lucky enough to begin collaborating with soon after I started to become a group leader at the EMBL in 2008. At that time, I'd known what cancer is, but I'd never had performed cancer research. And through interactions with him, we were essentially able to enter this field and learned a lot about concepts that are actually important to this field that relate to how variation is formed and how it is selected for, to go back to the main questions that my laboratory is addressing. And as you know, cancer is not purely but largely a disease of genetic variation arising in our genome. And in work that started as a collaboration with Stefan Pfister, my group was able to show that genetic variation in cancer can arise in big bursts, where much of the genome gets affected, seemingly affected at once. And I'm directly going into the data from a childhood brain tumor patient. The patient was suffering from a tumor termed medulloblastoma. Childhood brain tumors are actually a major threat to the life of infants. They're the most common cause of death in young children after deadly car accidents, if we consider the Western world, where infectious diseases and hunger are rarely going to threaten the life of a child. And we sequenced this medulloblastoma patient in my laboratory at EMBL and found that most of the chromosomes of this patient looked fairly normal when we sequenced the patient's tumor genome and the blood genome, and we plotted the data by comparing it to each other. So this flat line means not much going on this chromosome. However, a single chromosome, in this case chromosome 15, showed us evidence of a quite dramatic, seemingly explosion of events with over 200 breaks of the chromosome. And we soon noticed that this phenomenon that we stumbled across here had been recognized a few months earlier in a large number of cancer cell lines from cancer patients by Peter Campbell's group, and termed Chromothripsis. So given a Greek name for what we assume is happening here, that is the large-scale shattering of chromosomes and their rejoining. My laboratory found that Chromothripsis is specifically occurring in patients that had a very severe inherited mutation, a mutation in the TP53 tumor suppressor gene. And we could link both. And furthermore, we could show that in carriers of the TP53 mutation, Chromothripsis appears in this tumor type to have a nearly 100% penetrance, surprisingly high to us. And it also often leads to quite peculiar structures, small extra chromosomal structures, so-called double minute chromosomes forming, which we often find to contain more than one cancer gene. And this finding from my group and similar findings from Peter Campbell's group have led to the hypothesis that Chromothripsis can lead to the very fast evolution of a cancer by leading to more than one detrimental hit in the genome. In other words, more than one tumor suppressor or more than one oncogene being affected by the events. And in this case, we could verify the presence of this chromosome by fluorescence in situ hybridization and PCR. We're following this work, which is now a few years old. We've long been interested in what is actually the molecular mechanism behind Chromothripsis. And we wanted to take these results as a motivation to begin studying the molecular basis of how chromosomes can break into so many pieces which seemingly appears to happen at once. And a postdoc in my group, Balja Mardin, recently developed an experimental approach in vitro using a non-cancerous cell line to tackle this problem. And I want to go through this approach briefly. She uses for this RPE1 cells in which we depleted TP53, given the link of mutations in this gene with Chromothripsis. RPE1 cells are retinal cells that are immortalized, but they're not cancer cells. And she then perturbs these cells, either genetically or chemically, to force the cells over a strong selection barrier. She looks for cells that are able to grow in soft agar, which requires anchorage-independent growth, a characteristic that we only see in tumor cells. And she reasons that if she finds the right way to perturb the cells here, she might be able to see Chromothripsis events. That was the hypothesis. So did we get there? Before actually putting this approach to use, she needed to construct her TP53 depleted RPE1 cells, and she did so by using zinc finger nucleases. I'm very well aware of Emmanuel Charpentier speaking later. We performed this analysis before the CRISPR-Cas revolution, and used zinc finger nucleases. And when we did that, we noted that some cells became hyperploid, so these cells acquired a near tetraploid karyotype. And then we challenged the cells. And we used initially a chemical drug, doxorubicin, which induces double-strand break during the synthesis phase of cells, during the S phase. And we found that when we used doxorubicin, quite a number of cells transformed, were able to clonally grow in soft agar, and they showed characteristics of Chromothripsis. In this case, not quite as striking as in the medulloblastoma cell I presented to you earlier today, but according to current criteria, clearly Chromothripsis. We found additionally that Chromothripsis is specifically often occurring in cells that are near tetraploid, hyperploid, and Chromothripsis also occurs when we genetically perturb cells, specifically when we shorten their telomeres by using an siRNA against TRIF2, a component of the telomere-protecting shelterine complex. So with this, we are now able, in the laboratory, to regenerate Chromothripsis events, which is, for us, a prerequisite to study further this process, and by implicating hyperploidy and telomere shortening, we already provide a glimpse of how this method can be used to get further mechanistic insights that we would like to acquire in the future. And to get there, to understand Chromothripsis better, as we are a laboratory that also uses computational approaches, we're very keen on taking advantage of the large number of cancer genomes that have recently been generated in the context of the International Cancer Genome Consortium and in the context of the American component of it known as the TCGA. And since about 2.5 years, I'm jointly leading with colleagues in the UK, Canada, and the United States a project that is tackling the analysis of 2,800 complete cancer genomes, which we are jointly analyzing using an agreed upon set of computational approaches. So we're standardizing the analysis by using the exact same algorithms, and we need to perform much of this by cloud computing. At least we're doing this for patients where cloud computing is deemed to be an accepted approach, where we have the consent for this, to essentially save time and to be able to run the analysis we want to run within a reasonable time. There's also other data sets such as transcriptome and DNA methylation data available for this large set of cancers, which gives us further opportunities to learn about mechanisms by looking at the data in the right way. And I mentioned chromothripsis as one of the biological questions we would like to address by analyzing a large set of cancer genomes. So we can study chromothripsis in there and learn more about genetic mutations or epigenetic states linked to this phenomenon to follow up on the work on TP53, for instance. But there's also other aspects that interest us, that relate to the questions we are posing ourselves in the group, that is, how can we learn more about how structural variations are formed and selected for? And in this regard, I'd like to mention another discovery that we made recently again with Stefan Pfister and Robert Wexler-Rea from UCSD in California. Namely, we found that structure variations in cancer genomes often, when we looked at childhood brain tumors, often appear to affect regions in which no obvious gene could be identified, making us wonder why we would see a recurrence of structure variations in this case. To show an example here, in the vicinity but not directly affecting a gene known as GFI1, we find many structure variations in medulloblastoma that lead to translocations, exchanges between two chromosomes that appear to affect a region in chromosome 1, but which appear to partner with different chromosomes. And after further analysis, we found that what all of these alterations have in common is that they bring a DNA regulatory element near GFI1, in this case a lineage-specific enhancer element that is active in medulloblastoma cells. And through this, we uncovered the mechanism that we term enhancer hijacking to be a relatively abundant event in brain tumors. And since we now have 2,800 cancer genomes available, we were wondering, can we assess whether this mechanism of enhancer hijacking, where rearrangements in the cancer genome bring enhancers to new positions, is more prevalent? And to tackle this, we reasoned that it would be potentially a very sensible idea to integrate the latest knowledge on gene regulation, specifically how the 3D structure of chromatin predetermines which parts of the genome can interact with another. Using a method known as Hi-C, it is possible, namely, to understand which parts of the genome can interact or normally interact with each other in 3D space. These are the so-called topological association domains, or TADs, in which it is believed that most interactions between regulatory elements and genes take place. And for our research, we hence used the structure of topological association domains, and we asked the question whether structural variations in a cancer genome, here exemplified by this line, are often altering the structure of these topological association domains, which would allow enhancers that are normally not interacting with the gene of interest, here termed the non-cognate enhancer, to become a new cognate enhancer that can lead to an expression pattern that might lead to tumorigenesis then. Essentially leading to expression in a tissue where this expression would normally not happen. And hence, we developed a computational approach with these ingredients: the structure of topological association domains, or TADs, the clustering of structural variations in the genome, and information on where genes get expressed. And through this, we've identified 70 novel candidate loci where we believe that enhancer hijacking is actually the driver event of the cancer. Enhancer hijacking leads to genetic activation. And I'd like to exemplify this with one example here. A oncogenic locus in colorectal cancer, IGF-2, which we, following our approach, studied in more detail to understand the mechanism behind the gene activation. And I'm showing you here the genomic locus in which IGF-2 resides. IGF-2 is here in green, and we see that the structural alterations at the locus are very often duplicating IGF-2 once, or sometimes they're deleting a region downstream of IGF-2. The expression alteration we see in the presence of these variants is very striking. We see 300-fold overexpression for a single duplication and 100-fold overexpression for a nearby deletion, which argues against gene dosage effects driving the expression, quite strongly argues against it. And by performing chromatin studies, specifically chromatin immunoprecipitation followed by sequencing, we found that there's a very strong enhancer element in the vicinity of IGF-2, but separated by IGF-2 through the boundary of a topological association domain. So, genomically close, but distal from IGF-2 in a sense that normally no interaction between these elements would be mediated, we see a strong enhancer. This was a very interesting finding because the duplication we see here is typically a tandem duplication. Hence, the prediction would be that this duplication would carry both IGF-2, here again in green, and the enhancer here in purple, and bring them close by duplicating them, and possibly mediating the formation of a new TAD or contact domain that virtually only contains these two genomic elements. And that is a testable hypothesis that we pursued by a technique known as chromosome confirmation capture sequencing, which shows that in the presence of the tandem duplication, we indeed get very strong contacts here. And hence we can verify a computational approach in this sense, showing that indeed enhancer hijacking, in this case coupled by the formation of a contact domain, leads to overexpression. So this is one example of what we can learn by trying to understand how structure variations emerge and selected for in a large number of cancer genomes. And I'd like to, in the remaining 3 minutes or so that I'm kindly given, also relate to one additional type of analysis we are very interested to pursue in the context of the Pan-Cancer Project. And that is to link our knowledge on germline genetic variation and on what is happening in the cancer genome. So earlier I presented that TP53 mutations are linked to the formation of Chromothripsis in a brain tumor. I'm in the context of the Pan-Cancer Project, also abbreviated as PCAWG, leading the germline somatic association analysis, in which we are looking at both very rare mutations in high-risk genes such as BRCA1 and also more common mutations found in genome-wide association studies. And we're trying to understand, by looking at these inherited mutations, can we understand how somatic variation is formed and selected for? And as a prelude to this, we've studied in this regard, again, medulloblastoma, the cancer we're most comfortable with, looking at over 700 patients, and we found, first of all, that germline predisposition occurs in medulloblastoma much more often than was currently anticipated. Up to 30% of patients with a certain subtype of medulloblastoma have germline deficiencies in high-risk genes. Based on this finding, which is largely from a postdoc in my group, Sebastian Waschak, we're now suggesting new clinical care paths in medulloblastoma after subtyping and age matching. But we also, studying these genomes, made a very interesting observation. Namely, we've identified two genes, BRCA2 and PALB2, that previously in monoallelic form had not been linked to medulloblastoma yet. By looking at the abundance of carriers with these mutations in 700 medulloblastoma patients, and by comparing this to 54,000 normal individuals from a database of the Broad Institute. And we see here a log plot that shows us the expected versus observed p-values. These are the significant observations, which we only observe for mutations that have a high pathogenic consequence, such leading to a frame shift. But we don't see this for normal rare variants that are not predicted to affect the gene. So we identify two new factors that may be linked to medulloblastoma. Can we verify this linkage? And to verify this linkage, we looked at the tumor genomes of these patients. And we asked the question, in carriers of a PARB2 and BRCA2 mutation, can we, similar to what we previously seen in TP53, find any evidence of the tumor undergoing particular alterations? And we do see this. We see, as is shown here, a quite striking relative increase of mutations in these patients that carry BRCA2 or PARB2 deficiencies compared to sporadic medulloblastomas. We see an increased rate of somatic indels and a particular form of structure variation translocations, which strongly implicate homologous recombination DNA repair defects in these patients and make us comfortable with our finding that carriers of these variants are indeed predisposed to medulloblastoma. And I'm closing now by just reminding you what I presented to you today, insights into Chromothripsis, a striking pattern of genomic disruption that is linked to TP53, hyperploidization, and telomere damage, as well as approaches that we use to study cancer genomes, namely to understand enhancer hijacking and germline variation by linking germline and somatic genome. And I'd specifically like to acknowledge our collaborators and the people in my group that performed the work I present to you today: Sebastian Waschak, Joachim Weichenfeld, Balja Madin, and Tobias Rausch. Thank you for your attention.
Thank you very much. Merci, Jan. Elliot.
You showed an example of chromosome 17 undergoing chromothripsis in a patient. What happens to the other chromosomes? Do they all do it at the same time, or is it confined to single or subsets of chromosomes?
It is quite interestingly, we see sometimes more than one chromosome which appears to be affected by the exact same event. And we reason that because we then see that whatever results from chromothripsis is a connection of both chromosomes. So we see a derivative chromosome that contains shattered masses from two chromosomes, sometimes three. However, the majority of chromothripses affect a single chromosome only.
That's not the one in which TP53 is?
It is sometimes the one in which TP53 is present. Very often there's other chromosomes. In medulloblastoma, we appear to see an enrichment of chromosomes 2 being affected. And chromosome 2 harbors two important oncogenes for medulloblastoma, GLI2 and NMYC, which are then often affected by chromothripsis 2. So there's probably a selective advantage in chromothripsis on that chromosome.
Edith.
So over and above the amazing power of what you've been able to get thanks to genomics, I was just wondering whether you've started to explore epigenomics and the potential role of epimutations at both of the sort of levels you were talking about in terms of chromothripsis. Is there any correlation with the DNA methylation states of the genomes that undergo this? And could this be related to, for example, repetitive elements that become hypomethylated and therefore participate in this? So that's one level. And then the other is at the level of the chromosome organization and TADs, where some of these structural elements, like CTCF binding sites, are actually methylation sensitive. And it's been proposed that maybe epimutations could equally lead to enhancer hijacking or disruption.
Yes, certainly. So we are of course not an epigenetic lab, but we're getting closer. We're getting closer and closer to
genomics,
it.
You can do epigenomics.
Absolutely. So I agree with you. And I think this is a very interesting field, and you're posing very interesting questions that we're now getting much closer to because we're starting to look at data in a more integrative way. So enhancer hijacking, after we noticed that it was more of a surprise identification.
Right.
After we noticed that enhancer hijacking appears to be quite prevalent, we believe it may be as prevalent in some tumor types as fusion genes are, and people have long been
People have just
looking
been
for fusion genes,
on the genes,
but we've
not-
always
yeah.
been looking at cancer genomes in a narrow sense that we've looked for what we know already. So after we've seen that, we started to look, we should really start to understand where enhancers are present and where CTCF sites separate chromatin domains. So that's our entry point to epigenomics, if you so will. We've also looked much earlier in a very unbiased way. Do we see any correlation between methylation states and Chromothripsis?
Yeah.
We didn't see an obvious correlation, but that was 2 to 3 years ago. Maybe we should repeat it now with the pan-cancer data set.
And
I'm
also
sure
with
we
the
will.
hydroxymethylation, because it seems that that's the better correlate, 5-hydroxymethylation, than the CPG methylation.
That's a very good point.
Yeah.
In that case, we would be limited by the data sets we would have
Yeah.
accessible to us at large scale, but that wouldn't preclude us to work this out with collaborators that would have such data. And I agree, it's a very important question.